Next week, you will have the opportunity to get some bonus credit. I haven't decided whether it's going to be, how many points it'll be. It'll be at least 10 points, maybe as many as 20. I'm planning on giving you some problems, simple genetics problems to work on, and you can earn bonus credit by doing those and turning them in. They won't take too much of your time, so you don't have to worry about them until like Sunday after Thanksgiving, since next week is Thanksgiving break. But I figured it'd be a good chance for you to sit down and just work a few problems. Sorry. And that'll help you out. Technology. I always forget something. Okay, so there'll be that possibility, and also sometime between now and the last day of class, you'll be getting one more bonus quiz. All right, so there's the opportunity for points as well. All right, so we've looked through Chapter 9 up to a certain point. We've gotten the basics of Mendelian genetics, and we've looked at some of the modifications of Mendelian genetics that your book talks about. At this point, I'm going to skip back into Chapter 8 and look at cell division, and then we'll move back into Chapter 9 a little bit later to cover the rest of it. So Chapter 8 talks about cell division. You'll remember, hopefully, in the major points about these two chapters, there's two types of cell division, meiosis and mitosis. Mitosis is simple cell division where cells replace themselves. They make, basically, a cell divides into two identical copies of itself. And meiosis is the production of sex cells or gametes, where you produce cells that have only half the number of, or half the copies of genes, and therefore when you bring them back together again, you end up having a cell that has two copies of everything once again. All right, so cells have to divide for two reasons. They have to be able to replace themselves. That's where mitosis comes in. You have to be able to produce new cells for development. If you're going to develop a multicellular organism from a single cell, you've got to be able to divide. Those cells have to be able to differentiate to produce different kinds of tissues. So that's one reason that cells would have to divide. The second reason is that cells should be able to do, or organisms should be able to do, sexual reproduction. Okay, so to do that, you've got to half the number of genes, so that when those two gametes come back together, you've got two copies of every gene. Okay, we sort of get that from Mendel's rules. You remember Mendel proposed that these genes are present in pairs. They separate out so that each sex cell gets one or the other copy. Okay, so one copy of everything, and then when they come back together, you have two copies of everything again. So this is sort of mirroring or mimicking Mendelian genetics. All right, so some basic characteristics of cell division. One of the things that a cell has to be able to do is copy its genetic material. Right? That process is called replication. We won't look at the details of replication until next term when we do the molecular genetics component. But the basic idea here is that you have to be able to copy every single gene, all the genetic material, so that each daughter cell can get a copy in mitosis. Right? Cells have to be able to separate that genetic material appropriately. If you copy that genetic material, you don't want both copies going into the same cell. All right? The cells, when they divide, have to be able to distribute that material evenly. So that's where this aspect comes in. They have to separate that genetic material appropriately. When you produce sex cells, eggs and sperm, what you have to be able to do is separate that genetic material so that each daughter cell gets one copy of everything instead of two. Okay, so there has to be a way of separating all of those components. And you've got to be able to separate everything else. Usually this sort of happens by itself. Each daughter cell gets pieces of the rough ER, the smooth ER, the Golgi, all of those components. All right? So that usually happens without the cell having to do anything special. The mitochondria and chloroplasts in cells are separated out to daughter cells because those mitochondria and chloroplasts will divide independently of cell division. Right? So the mitochondria and chloroplasts are reproducing themselves while the cell is sort of doing its own thing. This is the simplest type of cell division. It's binary fission. Okay? Bacteria do this. A lot of single-celled organisms do this. It's just a situation where you copy the genetic material, distribute it evenly to daughter cells, and you produce two cells that are identical to the parent. So here's our parent cell. This is a bacterium. We've got this prokaryotic chromosome. You'll remember it's circular DNA. So it's a big circle of DNA that's sort of scrunched in on itself. And that's going to duplicate itself. They're going to replicate this, and then those duplicates will be separated as this cell wall grows. Okay, so what's really happening is more material is being added to the cell wall, and it's separating out that genetic material. Okay, so the cell grows in length. Once these two chromosomes are separated, then you get this furrow in the middle of the cells, and that will split the cells off so that each daughter cell ends up having the same genetic component that the parent cell had. These cells are basically identical to the original parent cell. All right, so that's the simplest type of cell division. For prokaryotes, the genetic material is a big circle of DNA where the ends are closed in on each other. It's fairly easy to work with that kind of material. You have the two circles that separate from each other, and that's it. With eukaryotes, it's much more difficult because the genetic material is composed of linear strands of DNA. And there's usually several different linear strands. Those are associated with proteins, and they're called chromosomes. Okay, chromosome just means colored body. When people were first looking at these cells and able to stain for various components in the cell, then they saw these funny things in dividing cells that looked like little X's, and they called them chromosomes because they were darkly staining. Right? Each chromosome is a long linear strand of DNA. We're going to sort of accept that in this class without formally proving it. You can prove it, but we do that in genetics. We don't have to worry about that here. Right? So each chromosome is a long linear strand of DNA, a single DNA duplex, okay, or double helix. Each chromosome has two ends. The ends are called telomeres. Okay, there's some funny structure about telomeres. The ends of the chromosomes are structured so that they don't fray or break down. And that's important because you could lose genetic information if those ends of those chromosomes break down. It turns out that most cell types can only divide for a certain number of divisions because every time they divide, they lose a little bit more from those chromosomal ends. Okay. It turns out also that cancer cells that are able to divide out of control and are able to invade other tissues have telomeres that repair themselves or replace themselves. So they don't have to worry about running out of DNA at the ends of the chromosomes. Okay? They're able to repair those ends. Each chromosome also has a constriction. We call it a centromere. That sort of implies it's in the middle of the chromosome, but it's not always. It can be very close to the end of the chromosome. Right? But that constriction is going to be important because that's how we're going to separate genetic material when we do cell division. All right, so telomeres and centromeres. When you duplicate chromosomes, those two duplicated strands are still held together. And those duplicated strands are called chromatids. I'll show you a figure that shows all these structures. Okay, so here's a figure that shows what we're talking about in terms of chromosome structure. Before the cell divides, you can't see chromosomes. They're really diffuse. They're all spread out in the nucleus, and you can't distinguish one chromosome from another. But if you could, you would think of them as a single strand. Okay, so here's our single strand. When that gets duplicated, the two duplicates are still held together at the constriction, or centromere. All right, so there's the centromere. We hold those together. And so these two independent strands, this one and this one, are called sister chromatids. They're sister chromatids because they're identical, theoretically. Okay. When a cell divides, what's going to happen is the strands, these two DNA molecules, are going to be separated at the centromere, and so each daughter cell gets a copy of that original chromosome. Right? So each daughter cell has the identical amount of genetic material that the parent cell had. Okay? So you see a single strand in those chromosomes. The strand duplicates, but they're held together at the centromere. And it's only in the later stages where these chromatids separate and are divided to the two daughter cells that are going to be produced. Okay. The ends of the chromosomes are here and here, of course. Those are the telomeres. You don't see the funny structure there because we're not drawing it, but it is an unusual structure. It's different than the rest of the chromosome. Cells have a cycle that they go through. Some cells will spend a lot of time in one particular phase of this cycle and less time in others. Some of them may never go into some of these other cycles. Okay, so our first cycle is growth phase, or gap 1 phase, or G1 phase it's usually called. Okay, that's when the cell is doing all the functions that it's supposed to be doing. If it's a liver cell, it's detoxifying substances. If it's a brain cell, it's producing signals that are going to other cells, that sort of thing. So whatever the cell is supposed to be doing, that's when it's doing it. There's a point in this cycle at which a cell decides whether it's going to divide or not. Okay? And if it does, if it gets past that point, then it is committed to dividing. Right? It can't stop, basically. Right? So that point, which is late in this G1 phase, that's the critical point. If the cell gets past that checkpoint, then it will go into S phase, which is the next phase. That's when the genetic material is copied. That's when the DNA is duplicated by this process of replication. Right? So G1 phase is the normal growth phase. That's when cells are doing everything they're supposed to be doing. Then they go into S. They copy their DNA. In G2, which is usually a lot shorter than G1, they're preparing to divide. Okay? So they're building up all the protein components necessary to do the cell division. And then finally, they go through cell division, either mitosis, as is shown here, or meiosis, if sex cells are being produced. There's two parts, really, of the cell division: karyokinesis and cytokinesis. All right, so what they're calling mitosis up here is really karyokinesis. That's division of the genetic material, or division of the nucleus. All right, cytokinesis then is division of the cytoplasm, and that happens late in the cell division process. Once those cells divide, then they're right back into G1, and they go through the whole cycle again. Actively dividing yeast cells, for example, will do this every 24 hours. Okay? If you have really fast-growing cells, they can do this in as little as an hour or so. Right? Some embryonic cells can duplicate all their DNA and go through this whole cell division cycle in 40 minutes, which is pretty astonishing. Okay, any questions on sort of the general information? What I want to do now is go into the details of mitosis, cell division to produce identical daughter cells. Okay? So two daughter cells are produced in mitosis. They have the same genetic material and same genetic component as the parent cell. They are identical to the parent cell. They're clones of the parent in a sense. Okay, this is done by what we call somatic cells. So that's a term you should remember. Somatic cells are non-gamete producing cells. All right, so any cell that's not producing eggs or sperm is a somatic cell. The cells in your muscles, the cells in most of your tissues, all of those cells are somatic. Okay. However, there are gonadal cells in very specific places, of course, that produce gametes, and those would go through meiosis instead. All right, so mitosis consists of 4 stages: prophase, metaphase, anaphase, and telophase, and there's very distinct things that happen in each one of those stages, or each one of those phases. Okay. All right, so what happens at each stage? What I want to do is go through each of these stages step by step, and then show you a picture that shows all of the things that are happening in the cell at that stage. All right? It really doesn't make sense until you actually visualize it. All right, so in prophase, first thing that happens, chromosomes start to scrunch up. They begin to condense. The way this happens is that they start associating with a whole bunch of proteins, and they get rolled up into these proteins. So that takes this big, long strand that may be millions of bases and scrunches it up into a very small package that the cell can easily manipulate or separate. All right, so that's one of the first things that you see happening in prophase. All right, the centrosome duplicates. You remember in Chapter 4, I hope you remember in Chapter 4, we talked about the centrosome as the portion of the cell, the part of the cell that makes microtubules, which are part of the cytoskeleton. Okay, the centrosome duplicates at the first of mitosis. And the centrosomes, duplicated centrosomes, then start to migrate away from each other to opposite poles of the cell. Okay? And as they do that, they're laying down microtubules. So you get this network of microtubules that's forming in the cell. Okay, so the centrosome duplicates, and they lay down microtubules. The nuclear envelope starts to break down. You can't get at the chromosomes and separate them unless you get them out of the nucleus. So the easiest way to do that is just break down the nuclear envelope so that the chromosomes are now out in the cytosol, the cytoplasm of the cell. Okay. Another interesting thing that happens in prophase is that you get these disks of protein that stick to the centromeres of chromosomes. Those disks of proteins are called kinetochores. Okay? And what's going to happen is that the microtubules from the centrosomes that are now at the opposite poles are going to attach to those kinetochores. Right? And so that's not only going to hold the chromosomes in the center of the cell, but it's also going to allow you to pull them away from each other simply by shortening the microtubules. All right, so here's some figures that show this process. These are amazing photographs. These photographs were taken by what's called a confocal fluorescence microscope. It takes a single plane image, and it's very, very precise and well focused. Right? So it's very easy to see structures. What's happened is all of these different components of the cell have been stained with different colors. And so in the nucleus we see this blue, which is DNA. All right? Here are microtubules, which are stained in green. And then the rest of the cytoskeleton is out here stained in red. All right, so in interphase, or that's the combination of G1, S, and G2, the chromosomes are very diffuse. It's very difficult to see them. Okay, so here's a diagrammatic representation of them. You can't distinguish chromosomes at that stage. All right, here's the centrosomes, and they've already duplicated. There's 2 copies of them, and they're sitting right here where all this mass of microtubules is. When you start into prophase, here's the centrosomes that are migrating now to opposite poles. And they're laying down microtubules between them. All right, so we see that here. You're getting microtubules starting to be laid down on either side of the nucleus. The chromosomes are starting to condense. You can kind of see that here. You're starting to see spaces in between these blue lines. That means that the chromosomes are starting to scrunch up. As prophase proceeds, the nuclear envelope breaks down. Right? So we see fragments of it here. The centrioles or centrosomes have migrated to the opposite poles, and you see the network or web of microtubules between them. And then here are the chromosomes, and they're well condensed. You can now distinguish individual chromosomes, these blue lines. Right? So everything's ready to get these chromosomes lined up so we can separate them. Okay, so in prophase, the centrosomes duplicate, lay down microtubules, the nuclear envelope breaks down, chromosomes condense, and they begin to line up at the center of the cell. All right, so in metaphase, the next stage of mitosis, chromosomes line up at the center of the cell. Why? Well, the reason is because you've got these microtubules extending from the poles of the cell. They're extending towards the center of the cell. They bind or attach to those kinetochores at the centriole, or the centromeres. Okay, so the microtubules are extending to the center of the cell. They attach to the kinetochores, and they cause those chromosomes to line up in a nice line right in the middle of the cell, which we call the metaphase plate. So once you've got the chromosomes lined up in the spindle, that is this network of microtubules, once you've got that fully formed, then you can separate the chromosomes, and that's what happens in anaphase. Right? In anaphase, we finally separate out all of those sister chromatids. They simply get pulled to opposite poles. Interestingly, the movement of these chromosomes is achieved by motor proteins. There are proteins that will actually walk along actin and microtubules, right, components of the cytoskeleton. So all you've got is these proteins that are walking on these microtubules and pulling the microtubules back. And when those microtubules get pulled back, so do the chromosomes. All right, so the chromatids are separated out to opposite poles. And then in telophase, that's where we do cytokinesis. Right? The nuclear envelope reforms, and the cells divide their cytoplasm. So you now have daughter cells. Okay? So that's the central furrow that divides those cells. So here's what it looks like. We've got our chromosomes lined up on the metaphase plate right here. Notice the microtubules that are extending towards the center of this cell. That's what's causing all these chromosomes to line up, because they're binding to those little kinetochores right at the centromeres. At this point, we can separate those chromatids, and so that's what happens. So here's all the chromatids that are being separated from each other, pulled and pushed to opposite poles. In this way, the daughter cell is getting the same amount of genetic material as the parent cell. Okay, 2 copies of everything. The nuclear envelope reforms in telophase. The microtubule network breaks down, and the chromosomes begin to decondense. They start to unravel so that they're very loose again. Okay, so prophase, you set up everything to be able to divide those chromosomes. In metaphase, you line them up. In anaphase, you pull them apart. And in telophase, you separate the cells so that they each have the same amount of genetic material as the parent cell had. And that's mitosis. All right, any questions on that? Good, okay. All right, meiosis is a much more complicated process, but it still involves lining chromosomes up and separating them so that daughter cells get the same amount of genetic material. Meiosis, though, involves two separate divisions, not just one. Okay, those gametes contain only half the genetic material. We'd say they have one copy of each chromosome instead of two. Okay, so in mitosis, the daughter cells have two copies of every chromosome. In meiosis, they only have one. Right? So that means that there's only one copy of each gene present. I haven't said explicitly that genes are on chromosomes. I might as well say that now. We'll assume that's so without formally proving it in this class, but there are lots of ways of demonstrating that genes are, in fact, on chromosomes. Okay, so here's a couple of chromosomes. And they're present in pairs in most organisms. Okay, if you have two copies of each gene and genes are on chromosomes, that means you must have two copies of each chromosome. Those copies are referred to as being diploid if there's both copies present. If there's only one copy, they're haploid. So there's where the terms haploid and diploid come from that are used in your book. Right, so one copy, haploid, two copies, diploid. Chromosomes that are pairs have the same genes in the same order, but they're not necessarily identical. They're not necessarily the same alleles. Right? And so in this figure, I've tried to demonstrate that. The left chromosome could be carrying a big A, and the right chromosome is carrying a little a. All right, so there's our big A's. Keep in mind that sister chromatids are duplicates of each other, so they're identical. All right, so these two sister chromatids are identical. You'd expect them to carry the same alleles. All right, we might have little a on the other chromosome. Okay? And again, these two are sisters, so they should be identical. Finally, another term, the location of a gene on a chromosome is called the locus of a gene. The plural is loci. Okay? Don't pronounce it loci. I hear a lot of people do that. Loci. All right, so if we were talking about the position of the A gene on this chromosome, we'd be referring to its locus on that chromosome. Its location on the chromosome. You could have other genes elsewhere on this chromosome. For example, you might have a little b up here and a big B over here. Right? Genes, there may be many genes on chromosomes. I mean, think about it. Humans have around 40 to 50,000 genes, but we only have 22 pairs of chromosomes, plus the sex chromosomes. Right? There's not one chromosome for every gene, so obviously you must have many genes on each chromosome. Okay, this figure sort of shows you how the different haploid and diploid states are derived during the life cycle of a typical organism. Right? These individuals are producing gametes, eggs, and sperm. Those are produced by meiosis, and those are haploid. In humans, that means that they have 23 chromosomes. Okay? When these unite in fertilization, they restore the diploid state. So you now have 46 chromosomes, two copies of each chromosome. Mitotic processes and developmental processes then give rise to the adult organism, which can then produce meiotic cells again. Okay, so there's a sort of alternation of haploid and diploid states in most organisms. If you used mitosis to produce gametes, right, if you use the standard mitosis process, you'd end up with gametes that had 2 copies of each chromosome. So if those fused, you'd then have an individual that had 4 copies. And so you can't do that. That's why you have to half the chromosome number in order to regenerate the original starting number in the adult organism. Okay, as I said earlier, there's 2 separate divisions in meiosis. The first division is called the reductional division, and that's the one where you half the chromosome number. Okay, you separate the paired chromosomes. In the second division, the equational division, you separate chromatids. Right, so important distinction. Meiosis 1, right, you separate pairs. Meiosis 2, you separate chromatids. Okay, so in a way, it's just like mitosis, because that's what you do in mitosis. You separate chromatids. Right, so you notice something that's different between this and mitosis. First of all, there's 2 divisions. Second, chromosomes are pairing in meiosis. They're not in mitosis. Very, very important distinction. If you remember that chromosomes pair up in meiosis, you'll never have any problem figuring out meiosis versus mitosis. Okay? Because you'll just pair those chromosomes and you'll immediately see what has to happen in the first meiotic division to separate those pairs. Okay, so it's a very important distinction. All right, so what happens in meiosis? Step by step, we're looking first of all before cell division. Again, the centrosomes have duplicated, so they're going to start migrating to opposite poles. We've got a nuclear envelope. We've got these sort of diffuse chromosomes in here. They haven't scrunched up yet, but they will as the cell goes into prophase. So as the cell begins prophase, the nuclear envelope breaks down, just like in mitosis. The chromosomes condense, just like in mitosis. All right? The centrosomes begin to migrate to opposite poles and lay down a network of microtubules. Same as mitosis. What's different? The only thing that's different is this right here. We've got chromosomes in pairs. The chromosomes have paired up. And in fact, those chromosomes will exchange pieces of themselves with each other. Okay? They cross over. That's what that's called. Okay? So we're indicating a couple of sites where these chromosomes have actually exchanged, physically exchanged pieces of themselves. They've crossed over. All right, so these pairs then in metaphase line up on the metaphase plate. Okay, and in the first meiotic division in anaphase, unlike mitosis, we separate pairs. All right, so you can think of these 2 fingers being paired chromosomes. They pair up. In the first meiotic division, you separate the pairs. Then in the second meiotic division, you separate the chromatids. Right? A real easy way to remember how that works. Pair up, separate pairs, separate chromatids. Right? You'll never go wrong with that. Now, look at another important thing. We started out here with 4 chromosomes. 1, 2, 3, 4. You can just count centromeres to know how many chromosomes there are. We separate those pairs in anaphase. Each daughter cell now has only 2 chromosomes. We've halved the number of chromosomes. We went from 4 to 2. So each daughter cell now has only one copy of each chromosome. That happens because the chromosomes are paired up and you separate pairs in the first meiotic division. It's why it's called the reductional division. We've reduced the number of chromosomes. All right, so this figure shows telophase of the first meiotic division, and it's a little bit easier to see the fact that we only have 2 copies or 2 chromosomes in each of these cells, whereas the starting cell had 4. Okay, 1 copy of each chromosome. What we have to do now in the second meiotic division is separate those chromatids from each other. And that's easy enough to do. You just do a division that looks very similar to mitosis. So we have our second prophase, where the centrioles duplicate, the nuclear envelope breaks down, the microtubule network is formed in the cell. Then in metaphase, the chromosomes line up on the metaphase plate. Right, so that's shown right here in the middle of this figure. Then in anaphase, you separate chromatids, just like you do in mitosis. Right, so the result of this is that you end up with 4 daughter cells, each with only 1 copy of each chromosome. Right, notice this is an equational division. We started out with 2 chromosomes. The daughter cells still have 2 chromosomes. We didn't reduce the number of chromosomes. That's why we call it the equational division. Okay, so in meiosis, you do 2 divisions. In the first division, chromosomes pair, the pairs separate. That halves the chromosome number. In the second division, the chromatids separate. Each cell still has half the original number of chromosomes, but you have 4 daughter cells as a result. All right, so here's a comparison of mitosis and meiosis. With this kind of a comparison, it's really easy to see the differences between the 2 types of cell division. Here's our starting cell where 2n equals 4. If you just count centromeres, you've got 4 chromosomes. Right? You replicate chromosomes so that you end up having duplicated chromatids. Right? Now you may be thinking, well, yeah, but didn't you just double the chromosome number? No, I doubled the chromatid number. It's an interesting distinction, but you just count centromeres whenever you're counting chromosomes. We've got 4 centromeres in here. We still have 4 centromeres. We've just duplicated those strands, and those strands are going to be donated to the daughter cells. So we still have 4 chromosomes. We just double the number of chromatids. In metaphase, there is no pairing in mitosis. These chromosomes just line up on the metaphase plate. You split the centromeres and divide those copies equally to the daughter cells so that each daughter cell ends up with the same number of chromosomes as the parent cell up here at the top. Okay, so both of these cells are still 2n, n being the number of chromosomes, haploid number of chromosomes. In meiosis, same thing is happening. We're replicating the chromosomes, but in this case, they pair. And they may cross over. So in metaphase 1, we have pairing. Right? That's really the thing that distinguishes mitosis from meiosis. You pair in meiosis and you don't in mitosis. So these pairs line up on the metaphase plate. You separate the pairs in anaphase 1. So these cells are now haploid. We start out with 4 chromosomes. Each of these cells now has 2. Okay, we have to do one more division to separate those chromatids. So we do a second meiotic division, which is basically just like mitosis. These chromosomes line up on the metaphase plate. They have nothing to pair with, so they don't pair. And you just separate chromatids. So you end up having 4 daughter cells that have half the number of chromosomes of the parent cell. Right? So from this figure, you can really see how to remember how meiosis and mitosis are different from each other. It's the pairing versus no pairing that's the issue. Okay? Chromosomes do not pair in mitosis. Chromosomes do pair in meiosis. Okay, any questions on those basic cell division processes? Okay. Interestingly, chromosome behavior in meiosis, the separation of chromosomes in meiosis, is very much like the segregation of traits in Mendelian genetics. Right? Mendel said that these genes are present in pairs. You've got a big A and a big A, for example, or a big A and a little a. And when you form gametes, the A's separate. So the gametes have one copy. Right? In a sense, that's the same thing that's happening here with chromosomes. When you separate chromosomes in meiosis, you get one copy of each chromosome. So Mendelian genetics is behaving just like chromosomes behave. And that connection was made as soon as people started observing chromosomes in the late 1800s. And they said, oh, well, the behavior of chromosomes looks a lot like the behavior of Mendelian genes. Okay, so, can the behavior of chromosomes account for segregation? And the answer is yes. Right? A related question, are genes on chromosomes? The answer again is yes, because chromosomes are the genetic material. Right? So genes are, in fact, on chromosomes. This gets back to that question I asked, a week and a half ago. How do we define a gene? We can't easily do it at this level, but we can at the molecular level. All right, so a couple of figures that will help you see how this works. There are two different possibilities for lining these chromosomes, these paired chromosomes up. Let's think of just this blue set versus the red set, although you could think of one set as the set that was obtained from the mother and the set that was obtained from the father, if you want to. Okay, so we have a blue set and a red set, and they can line up such that the blues are all on one side and the reds are on the other, or you can have blue, red, red, blue. Okay, those are equally likely. Either one is equally likely to occur. You've got a 50-50 shot. All right, so the arrangement of these chromosomes is pretty much random. Can either be this way or this way. So if it's this way, then what happens is in the first meiotic division, remember, you separate pairs. So those pairs separate, and we end up getting all the blue chromosomes in one daughter cell and all the red chromosomes in the other daughter cell. Those then divide, and we end up with two cells with all blue chromosomes, two cells with all red chromosomes. Right, those are two of the combinations that are possible. If we have this second alignment of chromosomes up here, then what happens is we get the large blue one and the small red one in one cell, and we get the large red one and the small blue one in the other cell. Right, so it's exactly the opposite arrangement. Then when you do the second meiotic division, it looks just like mitosis, you separate those chromatids, and we have these possible combinations. Right? Well, obviously you can't get both of those initial events in the same cell, but if you take a large pool or group of gametes, you would expect to see these combinations at the bottom of this figure in equal probabilities. Right? You have a 25% chance of having each of these four combinations. So far, that looks a lot like what Mendel said was going on. You remember that if you have, say, a big A, big B, little b, you separate those gametes so that you've got all possible combinations. Big A, big B, big A, little b, little a, big B, little a, little b. All equally likely. Right? Same thing is happening here. We have four combinations, all equally likely. Right? So what if we put alleles in? Does it work? All right, so here's an individual. We're going back to our round yellow seed phenotype in peas. This individual is big R, little r. The Y gene is on a different chromosome, so we've got big Y and little y on different chromosomes. In meiosis, we have two different possibilities or arrangements. We could have big R and big Y on one side, little r, little y on the other side, or the opposite arrangement. When you go through anaphase in these two possible arrangements, here you're going to end up with a cell that has big R, big Y, a cell that has little r, little y. Here's a cell that has little r, big Y, and a cell that has big R, little y. Come on. There we go. All right, so there's our cells. Big R, big Y, little r, little y, little r, big Y, big R, little y. We go through the second meiotic division, which is just like mitosis. You separate chromatids. You end up having 1/4 of the gametes being big R, big Y, 1/4 little r, little y, 1/4 little r, big Y, 1/4 big R, little y. Exactly as Mendel observed. Okay? So the fact that the chromosomes behave the same way that Mendel's genes behave is a strong suggestion that genes are on chromosomes, but it's not proof. The proof of that would have to come much later when people could sort of start working with individual pieces of chromosomes and playing with what they're doing. Right? And that's beyond the scope of this class. So we'll accept this, that this is sufficient demonstration to show that, in fact, genes are on chromosomes and that the behavior of chromosomes mimics the behavior of genes because of that fact. Right? So, of course, in the F2 generation, we're getting a 9:3 or 3:1 ratio, just as Mendel would have predicted. All right. Have to stop there, and we'll continue here on Monday. So I'll see you then.